Right, well, good morning, everybody, and welcome to the latest Pub MLST Forum event. And this is a special event in lots of different ways. Firstly, we are marking the 10th anniversary of the Meningitis Research Foundation Meningococcal Genome Library, and I'm very pleased to say that we've got in the room with us today Vinny and Linda, who from the MRF to mount that. And this is a hybrid meeting, so welcome to the new normal. I've got more people online, but we actually do have people here with us in the Martin School. So this is very much an experimental system. It's the first time we've done one of these things like this as a hybrid event, so bear with us as it goes. Just before we start, I'd just like to say a big thank you to Lucy, Andrea and Holly, who've done all the heavy lifting in organising this. Thank you very much. And also to Clara at the Martin School, who arranged the facilities. So everybody, I think, probably online knows that the MRF Genome Library is a open source library of meningococcal genomes that's been now running for 10 continuous years with whole genome sequencing of all of the meningococcal isolates obtained in the UK. So it's very much a first. It's based on the principles of open access, and it's embedded within the Pub MLST platform. So without more ado, I'm going to hand over to Chris Tang, who was really involved in the genesis of the Genome Library, and put this on present. Lucy. Yeah, oh, Chris.
Thank you, Martin. It's an absolute pleasure to be here and give the first opening address. And thank you, Martin, for organising the meeting. Thank you to MRF for sustaining and being involved in the Genome Library to start with. It's fantastic to have Vinny and Linda here, as Martin said. And I also want to reiterate thanks to Andrea, Holly, and Lucy, and my apologies as well for not sending slides on time. I thought turning up on time would allow me just to bring my memory stick in and shove it in the computer, but it's more complex than that, so I apologize for making it work late into the night. So I've been charged, following discussions with Jay and with Linda, with introducing the bacterium as a problem. And for everybody online, everybody here, I'm sure you understand, this is an organism which we know as a critical cause of sepsis and meningitis, particularly in childhood. Used to cause huge outbreaks in sub-Saharan Africa, within Europe and the US. Endemic disease, particularly by serogroup B, has been a problem for the last couple of decades. And the thing about the bacterium is at a biological level, is that it's specifically adapted for carriage in the human host. So we do know that humans are the only niche which this bacterium is found. Colonization is much more important than disease, or much more common than disease, with about 10% of the adult population carrying, although I think those numbers have been dropping based on recent carriage studies done by Martin and his group. But disease is very rare. And so the questions from that sort of standpoint, which immediately spring up, is why do some people come down with the disease? Are there certain characteristics in bacteria which make them more or less likely to cause disease? And how can we prevent it? So when the Meningococcal Genome Library was first thought of, just over a decade ago, there was no licensed vaccine against serogroup B, Neisseria meningitidis. So there was only the serogroup C vaccine, which was being used in the UK at that time. At that stage, the quadrivalent vaccine hadn't been introduced. So why does it cause such significant disease in individuals? Well, it's because the bacterium replicates to huge levels in the bloodstream. Tim Darton, working with Rob Reed, showed by RT-PCR that levels of bacteremia, if you measure bacteria by the number of copies of DNA in the circulation, can reach these massive numbers. And the bacterium proliferates, particularly in small vessels. This is a picture from a child that succumbed to bacterial meningitis on the right-hand side. And in red, you can see those small dots. Those are individual bacteria, essentially forming a colony, which occludes the microcirculation. And Xavier Nassif has seen similar results in his histopathology and in the model that he's been developing in transgenic, or in mice which have grafts of human tissue. And so we get large levels of bacteria in the bloodstream, and the bacteria release active substances, particularly lipopolysaccharide, which trigger immune responses. And it's really that sort of hyper elicitation of the immune response which is responsible for disease, and which led to the sort of real impetus for the Meningitis Research Foundation, for people working in the field, to develop vaccines to prevent this serious disease, which comes on extremely rapidly. And you'll hear later on in some of the talks about the importance of avoiding immune response, acquisition of nutrients. These characteristics are really essential for the bacterium to replicate to these very, very high levels. And so these have been interests in my lab as well as others. So if we just turn the clock back then to think when the MRF genome library really came to mind 10 years ago. As I said, there was no B vaccine. The vaccines which were under development, there was a vaccine from GSK at the time, which has turned into Bexsero. There was the Pfizer Trumember vaccine, which was under development. And so those vaccines were essentially based on a single organism without necessarily complete knowledge of the sequence variation of the antigens in the vaccines, and without complete knowledge necessarily, or fine detailed knowledge of the molecular epidemiology of the bacterium. So at the time, it seemed that some of the potential benefits of doing whole genome sequencing from a collection of strains, and the library really came about by a bequest from a donor, generous donor, to the MRF. I was approached by Chris Head, who was CEO of the MRF at the time, and Linda, to see how that money might be best spent. And it seemed that if we had a catalogue of genome sequences from strains which are carefully picked across the span of molecular diversity of Meningococcus, then we would gain a lot of valuable information about the bacterium in terms of vaccine development. And at the costing levels at the time that we thought of, it was about £1,000 per genome. We thought we might be able to get about 100 genomes done. But if we judiciously chose those in collaboration with people who understand things about population biology, then we might get interesting information. So in terms of vaccine development, we would really get a sense of the diversity of candidates, which might allow selection of specific antigens, specific variants of candidates. Obviously, the genomes contain all information of potential candidates, and then we'd be able to put that diversity into the context of the population structure. And I think you'll hear more about the population structure of Meningococcus later on. In terms of host pathogen interaction studies, the genome library would be able to generate hypotheses about what these disease strains had in common, and maybe they didn't share with carrier strains, so maybe understand why certain strains were more likely to cause disease. And it would allow us in the lab to choose relevant strains for our studies. And then finally, it might provide a really important platform for understanding the molecular epidemiology of the Meningococcus. And in that way, it would be able to be very easy to spot incoming new strains which were taking off within the country. So for all these reasons, we thought this would be an excellent idea to do to get a catalogue of about 100 strains. So contacted PHE, Ray Borrow, to see about the choice of strains, and fortunately discussed it with Martin as well, who emphasized that the existence of PubMLST and how that would be an ideal platform for the housing of the library. And in fact, I remember very vividly when we had a meeting at, I was reminded and remember very vividly about a meeting we had at Imperial at the time, when there was Linda, Chris, Martin, and Ray present. And Martin said, well, you don't have to do 100. You could do all of them, all the strains for the whole year. And in fact, what's happened since, it's a really pivotal, sorry about the impression, Martin. But but then it's been, to come up with the idea that you could actually do several hundred strains, really, expanded the possibilities with the meningococcal genome library. And the fantastic thing I think about the genome library, and it's a huge credit to those people who have been involved. That's people in Martin's group, people in Manchester, people at Sanger Centre, and the work now is taken on by Vinny and sustained by Linda, is the expansion of the genome library, not from England to Wales, but to include Scotland. Fact that it's been sustained now for 10 years instead of just the 1 year, which the bequest had originally funded. And now, the ambitions of the genome library is to go global. So we have a sense of the molecular epidemiology of this important pathogen, at a worldwide level. So this is a remarkable joint effort by the community. All of this is publicly available. I think we're incredibly fortunate as researchers to have benefited from the selfless work of the groups that contributed to this. And it's fantastic this is being converted, or thought of being converted, into how we can possibly eradicate this horrible pathogen from the planet. So it makes me extremely proud to have been originally involved in this, and I just want to congratulate people who've been involved in sustaining it and benefiting from it at this meeting. And with that, I think I will close.